Hello everyone, welcome to an academy. This is Ashwath. Today we will be discussing about cell cycle and division in this lesson. So, cell cycle and division is very important aspect of our life because since at the stage of embryo we keep on dividing and the cells of our body keep on dividing until we die. So generally the cell cycle and division process occurs in two types. First is called as mitosis and another one is called as meiosis. Okay, the cell can choose any way. It can divide through mitosis or it can divide through meiosis. That whole and soul depends upon the type of cells which is undergoing the division. Okay, starting with this initial phases of cell cycle, there are two phases called as interphase and M phase. M phase is also called as mitotic phase or mitosis. In the interphase, the cell prepares itself for undergoing a mitotic division or a meiotic division. Interphase has four phases: G1 phase, S phase, G2 phase and G0 phase. After the S phase and G2 phase, the cell can go directly to mitotic divisions or to meiotic divisions. In this video, we are discussing only about mitosis, not meiosis. That will be in the second part of this video. So, going into the topic, the interphase, as said, has four phases. In the G1 phase, it is an interval of time between mitosis and initiation of DNA replication. Okay, it is a phase between mitosis and initiation of DNA replication. So the cells is metabolically active and they grow, but DNA replication does not occur. Only initiation of replication occurs, but DNA replication does not occur in this G1 phase. In the S phase, DNA synthesis takes place. That means the size of DNA or the number of DNA doubles. If 2C is the DNA present in the initial phase, it becomes 4C. It can be denoted by 2C and 4C. It doubles. But the chromosome number remains same. If there are 2N chromosome set in G1 phase, then in S phase the number of chromosomes will be same or it will be diploid only. Coming to the G2 phase, in this phase proteins are synthesized in the preparation for mitosis where cell growth occurs or starts. Okay, this is the last phase which leads to initiation of mitosis. Some cells do not divide and they simply exit the phase. They simply exit the M phase. These are called as quiescent stage. This stage is called as quiescent stage. Coming to the M phase or mitotic phase, this mitotic phase is also called as equational division or equational process. This is because the number of chromosomes in the parent and progeny will be same even after division. So therefore this M phase or mitotic phase is called as equational division. This mitosis or M phase has four phases: prophase, metaphase, anaphase and telophase. We will discuss each of these phases. In the prophase, the DNA molecules which are formed during S and G2 phase, they are intervened initially. So what happens is, during the initiation of this phase, these chromosomes gets condensed. Suppose if the chromosomes or DNA present in the S and G2 are in this way, they become condensed in this phase. So the chromosomes start developing and they have two chromatids with a centromere. They have two chromatids with a centromere. This is centromere. They have two chromatids around the centromere. The initiation of mitotic spindle fibers, microtubules and protein components help for further processes in this phase. These mitotic spindle fibers and microtubules are secreted from the cell membrane place. Coming to the next phase, this is called as metaphase. During this phase, degeneration of nuclear envelope takes place. Okay, the further condensation of chromosomes takes place and gets completed. In this phase, two sister chromatids with a centromere are formed. Two sister chromatids with a centromere is formed. Centromere has a small structure on it called as kinetochores. Okay, the spindle fibers from the cell membrane attaches to the kinetochores as shown in this diagram, and all the chromosomes come to the center part or the equator part of the cell. This is called as equatorial region of the cell, or this is called as equatorial plate or metaphasic plate. During this metaphase, Golgi complex, endoplasmic reticulum and nuclear envelope disappears. The third phase of a mitotic cell division is anaphase. At the starting or onset of this anaphase, spindle fibers start to pull the chromosomes as center towards their respective poles. In the previous phase, these chromosomes were lying at the center of the cell. So in the anaphase, what happens is the spindle fibers, they attach to the chromosomes and pull the chromosomes towards their respective poles. Okay, the centromeres split due to this and the chromatids get separated. And the next and the final phase of mitosis is called as telophase. In this, the chromosomes reach their respective poles, okay, and the chromosomes further condenses again, forming a chromatin material. During this, the nuclear envelope comes around the clustered chromosomal material, and all the organelles which have disappeared earlier, like nucleolus, Golgi complex, endoplasmic reticulum, again they form in the cell. We can say that the mitosis starts with karyokinesis and ends with cytokinesis. Karyokinesis is nothing but separation of daughter chromosomes. Karyokinesis is nothing but separation of daughter chromosomes by division of nucleus, or in a cell division of nucleus occurs, then that stage is called as karyokinesis. In cytokinesis is a process where cytoplasm divides. Coming to significance of mitosis, why mitosis is important. It is also called as equational division, which is only restricted to diploid cells. So haploid cells do not undergo mitosis. Okay, the growth of multicellular organisms is only due to mitosis, and essential for cell to restore nucleocytoplasmic ratio. So mitosis is very essential for the cell to restore its nucleocytoplasmic ratio, and with the process of mitosis, cell repair can be done, or the diseased cells or the dead cells can be repaired. This is a diagrammatic representation of the early prophase and late prophase. In the early prophase, the chromatin material gets condensing, and finally chromatids are being formed in this late prophase. Coming to the next phase, this is after the late prophase, the chromatids keep on condensing and they form two sister chromatids with a centromere. Okay, during this early metaphase, the spindle fibers gets to attach to the centromeres or kinetochores, and they pull all these chromosomes to the center of the cell. This plate is called as metaphasic plate or equatorial plate. And the next stage is anaphase. During this anaphase, in the earlier diagram we have seen that all the chromosomes were in the central part. So the spindle fibers start to pull these chromosomes toward their respective poles. In the telophase, all the nuclear envelopes, the chromosomes again gets condensed or transits themselves into chromatin material, and all the nuclear envelope Golgi complex starts developing during this phase, and the cell divides. Okay. I hope you are clear with this concept, and we'll continue our mitotic phase of cell division in the next video. Thank you.